Welcome everyone to the PowerTalks series, and it's my pleasure to introduce Dr. Chang from Emory University. She's a new assistant professor of Biostatistics and Bioinformatics. Actually, her advisor is no strange to this crowd. 2 weeks ago, I believe 2 or 3 weeks ago, we had welcomed Professor Hongyu Zhao from Yale University to talk about statistical genetics. And her advisor is Professor Hongyu Zhao. But those of you who vividly remember the interesting discussion, and I did ask Hongyu Zhao about the novel statistical techniques beyond the polygenic risk scores. And I guess today, Dr. Su's work needs no introduction. She advanced from her advisor's Professor Zhao's work and ventured from statistical genetics into single cell analysis. And I think this is such an exciting area that the statistics and statistical method can still have tremendous impact in our field of bioinformatics. So without further ado, let's welcome her.
I'm going to take over. Thanks, Dr. Chang, for the very nice, very kind introduction. So I'm Chang Su, and this is actually my first time visiting a department as an assistant professor. So thanks very much to UAB. Thanks to Dr. J. Chen and Amy Wang for the invitation, and thanks to Amanda for scheduling the trip. So today I'm very excited to share with you my recent work. Actually, there are 2 papers on the statistical inference of cell type specific gene co-expression network with either single cell or bulk RNA-seq data. So feel free to interrupt if you have any questions. I think we should have plenty of time. Next one. Okay, so before starting, I would like to acknowledge this is a joint work with my PhD advisor, Dr. Hongyu Zhao at Yale University, my collaborator with Dr. Tingbei Zhang at Emory University, and also I would like to acknowledge the help from a couple lab members from the Zhao lab. So there will be 3 parts of the talk. Basically, first we cover what is cell type specific co-expression network, why we care about it. And next, we will introduce 2 statistical methods that we developed that use either single cell or bulk RNA sequencing data to estimate the cell type specific co-expression network. So these 2 methods are named CS4 and CSN. So first thing first, what is gene co-expression network? In this talk, I'm going to focus on gene co-expression that characterize correlation of gene expression levels across biological samples. So this type of network has been widely used in biomedical research to identify functional modules of genes and pathways. As a concrete example, here is an application or an example pipeline of co-expression analysis in the biology papers. So this figure is actually taken from a publication in 2013 to study Alzheimer's disease with microarray gene expression data, but I argued it is still quite popular, that this pipeline is still quite popular in both the transcriptomic and proteomic data analysis. So just to walk you through the pipeline, in this analysis, we usually start with some bulk tissue sample. In this case, they are studying Alzheimer's disease, so they collect some tissue from the brain. And then we use technology to profile either the gene expression or the protein expression in the tissue. In this case, they were using microarray to gather the RNA expression across samples. So with the measurement of thousands of proteins across hundreds of samples, we can start to characterize how the genes are correlated with each other and construct these co-expression networks. With the co-expression networks, a common downstream analysis is to do clustering. Basically, we try to group genes into small clusters that are highly correlated. And in this example, this heat map, the diagonal, we can see a couple of red blocks here indicating the genes that are highly correlated with each other. And if we do some functional enrichment analysis, we usually find some interesting biological function carried by these gene modules. In this case, they find some gene modules on astrocytes, unfolded protein, or immunity, which are all very relevant to the pathogenesis of Alzheimer's disease. And as a final step, people will do this association analysis to associate the gene modules identified from co-expression network with either genotype to understand the genetic regulation of the function, or associate with phenotype to understand how this function might be dysregulated in, say, Alzheimer's disease patients versus control. So this pipeline has been quite powerful and popular in RNA-seq world in analyzing the gene expression data. And it has been able to offer these functional and mechanistic insights into tissues and diseases. And specifically, it takes a system biology approach by grouping genes together into the units of biological function. And this function can be used to correlate with different phenotypes or genotypes to generate testable hypotheses. And finally, the relationship between genotype and these functions can also help us to understand genetic effects in context. So despite the great advantage and application of this pipeline, there actually exists some major limitation with this analysis, being that the data are collected at bulk tissue level, which are actually composed of many functionally distinct cell types. Taking brain as an example, in the brain tissue, there are actually all these different cell types. You have neurons, astrocytes, microglia, oligodendrocyte. They are all different cell types with different biological function, and as a result, different co-expression network in these cell types. However, using solely the data from bulk tissue and where every cells have been mixed together, we don't have the resolution to reach the cell type specific co-expression network and biological functions. So to be a little bit more concrete about why this could be a challenge or a limitation, first, if you estimate any gene pair to be co-expressed using the bulk data, it is unclear in which cell type do these two genes co-express or work with each other. And secondly, even more concerning is that the bulk co-expression that we observe could be actually an artifact due to the confounding by cell type composition. So to be clear about what we mean, let's consider a toy example. Say we have a gene pair that are only expressed in cell type 1, but they are not co-expressed with each other. So as this toy example, we can see gene 1 and gene 2, they both have expression level of 10. And if we zoom in, there are indeed no correlation between these two genes, indicating that they are not co-expressed with each other. And these are only the cell type specific expression. But in order to get a bulk expression where all the cell types have been mixed together, we further hypothesize that there are 20% of cells from cell type 1 in this bulk sample. And so we recalculate this normalized expression level at the bulk level, and we can see that the expression level go from 10 to 10 times 20% to 2, because this is a gene pair that are only expressed in cell type 1, and you only have 20% of cells from cell type 1. So everything looks good for now. These gene pairs, they are only expressed in cell type 1. They are not co-expressed with each other. And let's consider an additional setting where we have another set of bulk RNA-seq sample, and everything is the same. These two genes, the cell type specific expression level are the same. They are not co-expressed. But the only thing different is that this second set of samples now have much more cells from cell type 1. So we can see that, again, similar expression level in the cell type specific picture, but now we have more expression in the bulk picture because we have more cells from cell type 1. So looking at these two sets of bulk RNA-seq sample, like nothing's wrong, like we have these two genes with no co-expression. But if you mix them together, which is exactly the case in real data, where you have this heterogeneity in cell type proportion across the population, and you again plot their gene expression against each other, we will see a very high correlation between these two genes that are actually caused by the confounding effect of cell type composition. So this is what we mean by the bulk co-expression could result as an artifact by heterogeneity in cell type composition. So hopefully I have convinced you at this point that existing co-expression analysis could be limited in terms of if we observe a gene pair to be co-expressed, we don't know in which cell type do they co-express. And it is also possible they are not co-expressed at all and arise purely as an effect, as a result of the confounding effect. So we argue that it is possible that this bulk estimate may fail to capture co-expression in specific cell types. So to address this limitation, to address this challenge, we propose two new statistical methods. The first one called CSNet, where we aim to use the bulk RNA sequencing data where cells have been mixed together to infer these colorful cell type specific gene co-expression networks in the middle. And the second work we developed is to use single cell RNA-seq data where cells have been profiled individually and can be classified into cell type. And we developed this tool called CS4 that infer these cell type specific co-expression networks. So both of this work has been recently published. The single cell one is published in Nature Communication last year, and the bulk RNA-seq one is published in JASA this year. So in this talk, I will focusing on the work that use single cell to infer gene expression network, but also briefly touch upon the second one. Okay, I can pause here a little bit if there is any question.
It's no question, just a comment. It's good you brought up the single cell versus our bulk RNA-seq because it's very important to differentiate between the two.
So I have a question for this tool. Is it gonna be analyzing using both bulk and single cell RNA, or if I only have the single cell RNA level, I can still using the tool?
Yeah, if I understand you correctly, you are asking about the integrative analysis of both single cell and bulk data for inferring the network.
So is this for that purpose, or is like the CSNet only for the bulk and the CS4 only for the single cell?
Right. It's like respectively.
Okay.
Yeah, we developed one tool for bulk RNA-seq, one tool for single cell, but it would be very interesting if we can develop a tool that integrates two data sources to estimate the same thing. It sounds like a natural next step, but I haven't started working on it. Okay. Yeah, there's no more question. So I will go into the first tool we developed, which is a CS4 that uses single cell RNA-seq data to estimate cell type specific co-expression network. So the idea is pretty straightforward. In single cell RNA sequencing, you have all the transcriptome of each individual single cell, and you have a lot of tools that allow you to annotate the cell type and group cells into cell types. And maybe for cells of the same cell type, we can just apply some statistical tools to infer the cell type specific co-expression network. However, this is actually pretty challenging due to some characteristic of single cell data. So just in order to describe the challenges, I will first describe the new terminology. So in the most popular pipeline right now, the drug lab-based single cell RNA sequencing data, we use unique molecular identifier count, the UMI count, to quantify basically the expression level of genes in single cells. The larger the count, the more highly expressed the gene is. The other concept is called sequencing depth, where we sum up all the counts detected for all the genes in each cell and call it sequencing depth. Basically, it's a notion of how much information you have for each cell in your data. So the unique challenges with single cell data for inferring co-expression network is twofold. The first, we have the sequencing depth variation that are highly variable across cells. It can vary from hundreds to tens of thousands. And as a result, we will see it can actually confound the co-expression relationship between any two genes. And a lot of existing methods can yield inflated false positive due to the sequencing depth variation. And the second challenge we are going to describe is the measurement error, Where these UMI counts, they are a noisy measurement of the underlying mRNA abundance level. They are not perfect. And this error, as a result, will attenuate the co-expression relationship between any two genes and will also show more results. So before we show any numerical results, here are some benchmarking we did. Basically, in all the analysis we did, we compare our new method, CS4, to a lot of the existing method, either the generic method, like the classical Pearson and Spearman correlation that applied on more normalized data, or a couple of methods that have been specifically developed for handling single cell data and for inferring co-expression network with single cell data. So these are comprehensive references. And we will contrast their performance in handling those two challenges with our newly developed method CS4. So to go into the first challenge, what do we mean by confounding by sequencing declaration? So here I illustrate some cells and I order them by the magnitude of their sequencing depth from the left to the right, so that we can really see the sequencing depth really varies a lot from hundreds to tens of thousands in real data. These are real data. After visualizing the sequencing depth, I would also like to show you what the data might look like. So here I took some genes in these cells and visualized the UMI count with a heat map. So each individual block here is the UMI count detected for one gene in one cell, and the redder the color, the more counts you are detecting. So by eyeballing and staring at this matrix, you might notice two things. The first is that this matrix is very white, in the sense that it is highly sparse. A lot of the entries are zero. So this is what you get for real single-cell RNA-seq data. It is highly sparse, over 90% zero. And the second feature I want you to pay attention to is that this matrix is always redder towards the right-hand side, meaning that you always detect more counts for any genes in cells that are sequenced more deeply. So this is actually a common feature of single cell or of any, I think, sequencing data. If you sequence more deeply, you will get more things. But what it means for co-expression is that if you just pull any two genes and plot their count against each other, they will likely appear to be highly correlated, due to the fact that any two random genes will always have more counts simultaneously in cells that are sequenced more deeply, regardless of their true co-expression status. So this is what we mean by confounding by sequencing declaration. This great variation in sequencing depth, if left unaddressed, can introduce many false positive correlation between genes. Even though this might seem like a naive feature of the data, but we actually found in real data experiment that a lot of the existing methods are prone to such confounding effect. So to demonstrate this point, we did a permutation experiment where we randomly permute the gene expression across cells for all the genes, such that any two genes should be decorrelated and have co-expression equal to 0. And we then test different methods on their performance under varying sequencing depth versus constant sequencing depth. So these are the performance of 8 existing methods. On the y-axis is the scaled average co-expression estimated by each method. And varying, the red color denotes the scenario where you have varying sequencing depth and potentially confounding effect from sequencing depth. So we can see that when there exists varying sequencing depth, a lot of the existing methods give you false positive or highly inflated co-expression estimates, where they estimate independent gene pair to be correlated. In contrast, when you specify constant sequencing depth in this experiment, a lot of this inflation goes away. And this contrast between the red and blue curve shows you that it is the confounding, it is the variation in sequencing depth that caused the confounding in the performance of these existing methods. In contrast, we also apply our method in these settings, and we found that our method is pretty robust to the varying sequencing depth, indicating that we can handle the confounding due to sequencing depth pretty well in our methods. So hopefully I have convinced you that confounding is a big issue and CS4 can handle the confounding. And next I will move towards the second challenge. So now we just show challenges that sequencing variation confound existing method, we are robust. And we focus on the next challenge, which is the measurement error. We will show that existing methods tend to be attenuated by measurement error, where our estimates remain unbiased across expression methods. So to see what we mean, again, consider a naive, just a toy example, where hypothetically, suppose we can observe the underlying gene expression of two genes. And we know that the underlying, like the mRNA molecules in the cell, are correlated at a correlation 0.9. However, in real data, we don't get to directly observe the mRNA molecules perfectly, but rather we measure them with the noisy UMI counts. So after convoluted with the measurement error, we can see that the correlation between these two genes gets much more attenuated due to this additional error introduced in the sequencing step. And to see how existing method may be affected by this challenge, we again do a simulation experiment where we simulate the true gene expression level. We simulate the true co-expression to be 0.5, the cross-expression level of the gene pairs. And we see that a lot of the existing methods yield downwardly biased estimates of this gene pair, while our method remains robust across different gene expression levels. So at this point, you might be wondering why the new method CS4 performs so well against both confounding and attenuation. So the key reason lies in the careful modeling of single-cell RNA-seq data. So just to introduce our model, this is a model that we borrow. It's called expression measurement model, proposed in a Nature, I think, genetics paper, where they propose to use this expression measurement model to disentangle the two sources of variation in single cell data. Where for cell I and gene J, we use the DIJ to characterize the underlying biological variation, that is the true underlying gene expression level, how they varies across cells. And we have a second component that is dedicated to the measurement step, that is sequencing step, or the step where noises are introduced. And we use a Poisson model that we model the observed UMI count conditional on the underlying gene expression to be following a Poisson distribution. And here we specifically introduce the sequencing step here, which is shown to be beneficial later on in controlling the confounding due to sequencing variation. So in this classical model, a lot of the times what people do is to use some likelihood-based approach for estimation and inference. Say we are interested in either the underlying mean expression level or the co-expression between genes. People will do some likelihood assumption where we assume this underlying gene expression level to be gamma distribution, such as marginally Xij will be a negative binomial distribution, and we can just write down the likelihood, optimize it to estimate the unknown parameters. However, we argue that this approach can be quite challenging to work with, or it doesn't even work to estimate a network. So the previous model was proposed for a single gene, but in the case of estimating the network and handling multiple genes, you will need to deal with the multivariate gamma distribution, which is hard to even define. There are multiple versions of it. And it is very, very slow to fit this likelihood-based algorithm, and they are known to be suffered from convergence issues. So in our work, we propose a moment-based approach called CS4 that puts no distributional assumption on the underlying gene expression levels, and it is very fast in terms of how we can take less than 10 seconds to fit a large network, and it also offers statistical tests with theoretical justifications. So just to go in a little bit of detail, so this is the expression measurement model that we use in CS4, where we jointly model the P genes altogether with a general distribution FP without specifying any parametric assumption. We just assume it to be an unknown non-negative p-value distribution. And again, for the measurement step, we use the commonly used Poisson assumption. And we define the co-expression to be the correlation of the underlying gene expression level, which is by definition not related to measurement error and not confounded by sequencing step, which are both introduced in the later steps. And in terms of the real estimation and inference, we leverage the moment condition that can be derived based on the model we just showed. So for the expression level, we can show the mean variance and covariance. And this is how we define the mean and variance and covariance between gene J and J prime. And given this definition and notation on the underlying gene expression level, we can further write down the moment condition for the observed count. And we can see the moment condition on the observed count connect nicely with the moment condition on the underlying gene expression level. So we can see this is the regular Poisson expectation. This is the variance term here is a Poisson component plus sum over dispersion. And finally, we can also connect the observed covariance with the underlying covariance of the underlying gene expression level between two genes. So given this framework, we further set out to estimate this unknown parameter, which is the mean expression level, the variance of each gene, and the covariance of each gene using a moment-based regression approach. Basically, we match the sample moment, say the xij equal to x expectation, plus some random error. And we think of it as a regression where x is your observed UML count, si is your covariate, and mu j is the unknown parameter to be estimated. Similarly, we can write down this equation for the variance and the covariance terms. So we can sequentially estimate the unknown parameter mu j, sigma j prime as the estimates for this underlying gene expressions distribution. And just a little bit on the statistical technique, we use something called the iteratively weighted least square to achieve efficient estimation. Basically, the regression approach we showed just now, we didn't characterize the variance in these random errors. But if we characterize that carefully and carefully weight our observation when we do the estimation, we can obtain more efficient estimate, that is estimate with less variance. Yeah, so finally, aside from estimation, we also propose a statistical test that allows us to test for the independence between any two genes. So we here, based on the regression approach, basically, and the iteratively weighted idea, we can derive a asymptotic test that is well justified theoretically and also perform quite well in real data analysis. So that will be all on the method part of CS4. If there is no more question, if there's any question, I can take any question. Okay, yeah, so let's move on. So next will be the more exciting part, which is the systematic evaluation and real data application of CS4 in to understand biology. So first, we did some benchmarking experiments on how accurate it is, how fast it is, and also how it performs in some complement downstream analysis. I keep going back. So this slide is on how the accuracy and speed of CS4. So in this slide, we do some simulation experiments, and we can see that CS4 is the method that gives the best recovery of the true network. And also, we compare the speed of CS4 against a lot of other methods on the log scale. So we can see that it really performs, it calculates very fast and even comparable to the classical Pearson and Spearman correlation. And then we also systematically evaluated on real data. We focus on two disease phenotypes. One is Alzheimer's disease and the other is COVID-19, where we look at the functional enrichment of the identified gene sets and gene networks, and the reproducibility across data set, and how the identified gene pair are consistent with public databases. So this is an overview of all the data sets we used. So this is an example where this is the first real data example where we apply the CS4 method to the microglia cells in the brain. And we try to identify biological functions that work like that were critical to microglia. So in this case, we pick some GO functions that are known to be relevant to microglia's functions, say the chemical synaptic transmission, the protein folding, defense response, and we estimate the network with either CS4 or the second best performer a method called rho-SC transform. in our benchmarking experiment. So it performs the second to the best in all the evaluation tasks. And we compare the network estimated using either our approach versus the second best performer. So we can really see that CS4 in this case can accurately identify the strong correlation between genes in the same GO pathway and also distinguish between these GO pathways. While the rho SC transform, the estimates are pretty attenuated as what we just described, and it cannot discover or uncover this relationship between known pathways. And we further do some validation analysis where we validate our computational estimates by some numerical evidence. So the first experiment we did is to grab two different completely independent data sets, estimate the same network, and evaluate how consistent or how much reproducibility we can get when comparing these two computational estimates on two different data sets. And again, we compare CS4 to the second best performer in our benchmarking experiments. And the larger the ratio, it means that the more reproduced we are compared to the other methods. And we can see that across different p-value cutoff and across different brain cell type, CS4 can achieve better reproducibility on the identified gene pairs. And the second experiment, the idea is similar, but now instead of comparing with a orthogonal data set and computing another set of estimate here, we compare our estimates with a known database on transcription factor target genes. So some known gene pair that are known to be working with each other. And again, we evaluate this reproducibility or the consistency ratio and see that our method consistently gives more biological findings that are more coherent with the existing knowledge. And finally, it's an application on studying the disease mechanism. So in this application, we use, we used CS4 to study COVID-19, where we focus on the monocyte, the cell type monocyte, and we apply CS4 to the control cells and COVID-19 cells separately. And we analyze the differential co-expression between these groups, namely which gene sets work with each other in the most different way in disease versus control. So in this case, we find some really interesting differential co-expression between genes. And say in the COVID-19 sample, we can see that a lot of the interferon stimulated genes, SARS-CoV-2 related pathway genes are found to be strongly upregulated, and the co-expression is found to be much stronger in the COVID-19 cells compared to control, which is consistent with our existing knowledge about the disease mechanisms and pathogenesis of COVID-19. So this is other results I have for the first work, CS4, that use a general expression measurement model to provide unbiased estimates, good possibility control against confounding. And we also offer accurate downstream analysis as well as reproducible and meaningful biological discovery across cell type, tissues, and diseases. So that will be all on the first part. So that's all with using single cell data for co-expression network estimation. In our other project, we also tackle a different task, which is to use the rich collection of existing resource of bulk RNA-seq data to recover these cell type specific components. So why do we want to use bulk data? The reason is that bulk data, even though it's a little bit outdated now, but it's lower cost to collect, and there exists a lot of bulk data that have been accumulated over the past 15 years. And the idea we are going to describe is actually pretty close to the deconvolution literature that is popular in the bulk RNA-seq field in recent years, where we try to deconvolute the bulk data to gain cell type specific insights. So in this project, we also aim to do a similar task that leverage bulk data and deconvolute to get cell type specific co-expression network. And this will be equivalent in the statistical language, which is to estimate large covariance matrices from a convolution of densities. So in this second project, we take a more statistical flavor where we carefully model the bulk gene expression observation. So assume we have N samples, P genes, and K cell types. We denote the bulk gene expression with Xi, we denote the cell type specific proportion and expression with high K and SIK. So we assume a pretty simple model on the observed bulk gene expression, where this observed bulk is a weighted sum of the cell type specific gene expression with weight given by cell type proportion. So even though this model is simple, it's not trivial to estimate the covariance matrices from based on this model. But we can first write down the moment condition. So again, if you put an expectation or a covariance on the left, you can derive correspondingly how, say, the bulk expression level is composed of cell type specific expression level, or how the covariance matrices is actually a weighted sum of the cell type specific covariance matrices. So the weight here separately is given by pi ik for the mean and pi ik squared for the covariance, which makes sense because covariance is the second order. And so given this notation, this is basically just a graphical illustration of the equation that we just showed. We model this covariance matrix of the bulk data, denoted as the gray matrix here, as a weighted sum of the cell type specific components with weights given by cell type proportion. And the task of our project is to infer these colorful components from what we observe, which is the gray component, the sample covariance matrix of the bulk expression levels. So how should we do this? The first idea we had is also to resort to the more likelihood approach to optimize some likelihood to get the unknown parameters. But this turns out to be quite challenging because we are dealing with a convolution of densities. So just to illustrate the point, if we put a simple classical multivariate Gaussian distribution on the underlying gene expression level xik, so for cell type K, we can write down the log likelihood for the observed data, which is found to be not convex or biconvex with respect to the unknown parameter, which is the cell type specific co-expression covariances. As a result, likelihood approach may not work very straightforwardly in this setting. So again, we propose in a similar flavor as CS4, which is a novel moment-based approach, that instead of focusing on likelihood and optimizing the likelihood, we focus on the moments where for each gene pair, we know that the covariance, so the expectation of the second moment, is a weighted sum of the cell type specific covariances with weights given by cell type proportion. And we can again apply the moment-based regression idea where we match the sample movement. Say this is the equivalence of this is the sample equivalence of the covariance. It is equal to the mean parameter here plus some random error. So we can think of it as a regression where this is the unknown, this is the observed response variable. The pi square is the covariate, your usual x in regression. And sigma jj prime k is the unknown parameter, the beta to be estimated, while epsilon is the commonly seen, the random errors. So we propose to apply such a moment-based regression approach, where we first element-wise estimate these cell type specific mean or covariances using the weighted least square estimator for these components. And we can compute the co-expression matrix, which is the correlation. And in step 2, we further propose to do some thresholding when the number of genes is large. The reason we add thresholding here is because in both RNA-seq sample, you usually have hundreds in terms of sample size, but the number of genes can be huge. So in this case, we need something to handle the high dimensionality of the data. So we propose to do an element-wise scattered thresholding in large P setting that gives us nice theoretical results. So just to give a flavor on the main results we got in this paper, so we proved the non-asymptotic convergence rate as well as the selection consistency of the CSNet estimator under certain regularity conditions. And we also done extensive simulation results to demonstrate that the CSNet estimator can recover the true underlying cell type specific covariance matrices. And finally, we have been, I think, pretty good application to Alzheimer's disease. So again, we focus on AD and we use this a large cohort on Alzheimer's disease, the ROSEMap study. And there have about 541 whole RNA-seq samples that are collected from human brains, and the data have been normalized with FTKM. And we focus on 61 Alzheimer's disease risk genes, and we want to understand how these genes work with each other in a cell type specific context. And one thing you might have noticed is that all our regression proposed in CSNet, it actually requires the knowledge of cell type proportion. So we use an external tool, which is the CyberFlow-X published in Nature Biotech in 2019. So we use it with some external reference data to get the cell type proportion estimate, which are used as the covariate in our regression. So here are some results from applying CSNet. So on the left picture is what you will get if you just calculate the sample correlation of between genes using the bulk data. And we apply CSNet both before and after thresholding to study the cell type specific picture of these co-expression matrices. So in this case, we can see a couple of things. So say in astrocyte and microglia, we can find these couple of small clusters that seems to be the strongest in the astrocyte cell type, or this middle red cluster that seems to be having a stronger signal in the microglia cell type. And going into the literature for this this small cluster that are identified to be highly co-expressing astrocyte, we found that it has this famous Alzheimer's disease gene, the ApoE, AD risk genes, and it is known to be primarily expressed and produced in astrocyte. Also, it has this gene pair ApoE and ABCA7, which are known to be involved in lipid metabolism and phagocytosis, specifically in astrocyte. So we identify this cell type specific co-expression that are consistent with existing knowledge of the brain cell type. And one more example is on microglia. So actually this middle cluster, even though a little bit noisy, but we observe the strongest signal primarily in microglia. And these genes, when we look up the gene names in the literature, we found that a lot of these genes are known to be involved in AD etiology via microglia. And also it is known to, a lot of them are known to be uniquely expressed in microglia. So this example really demonstrates that we can refine and get this cell type specific insights on this co-expression network and really filter down and find this cell type that is the origin of the co-expression between two genes. Yeah, so just in summary, in CSNet, we develop a deconvolution network that deconvolute the observed bulk data into the cell type specific component for co-expression network estimation. And we offer theoretical guarantee for this method, which is, and the method is also flexible, computationally efficient, and it was able to generate biological insights on disease mechanisms. So just to summary that this is the two paper that we have on CSNet, CS4. I think CS4, we provide both Python and R version in terms of implementation. And for CSNet, we have an R package and everything is reproducible. All the results in these two papers are reproducible. So I'm happy to take any question. Yeah, I am happy to take any questions for these two methods. Thank you.
Well, let's open up the mic if you have questions. I can try to start. So for your application to Alzheimer's disease, did you find anything interesting more than the existing knowledge that we all know? Like,
yeah, that's a short question. So in the paper, when we do the application, we were excited by finding things that we already know. But for things that are novel, I think we will need to work with collaborators to really confirm that in downstream further validation experiments, but we haven't done that yet.
Hi, I'm Zhong Yue from Emory University. I have a question. So have you considered about the multi-omic data, like the GWAS data, that to be important in your co-expression network as well, or some proteomic data as well? Thank you.
Yeah, that's a good one. I haven't considered how GWAS can be incorporated, but I imagine, say, GWAS disease risk could be enriched in certain biological pathways, say the gene module or gene subnetwork. That could be complete. That could be very possible, but I don't have a good idea in terms of how to integrate that information for estimating network. But on your second point, proteomic, yeah, I'm actually working on it right now to apply the CSNet method to a large proteomic data we have at Emory. So we also have many, I think a couple of hundreds of proteomic data from the brain on Alzheimer's disease patients. But the question there is that it's similar to RNA-seq. The data is not cell type specific. It's not at single cell level. So we are trying to figure out a way to see if we can estimate the cell type specific.
Thank you.
Hey, I have one question about the CS score. So a major limitation in generally in co-expression network analysis is the number of samples that you have. And being a single cell, so we work with single cell core, and we do see that folks generally have 2 or 3 samples per group. And then it becomes a limitation to perform any co-expression network analysis or some kind of analysis, because you will get a spurious correlation because the sample size is so low. So have you tested your method in any of the single cell data which have a very restricted number of samples?
Okay. So just let me say the question back to make sure I understand. So the question is about sample size and how we do network estimation when there's very, very limited samples.
Yeah, that's true. That's the question.
Yeah, I think 2 or 3 samples for thousands of genes, that will be very, very challenging, or if not impossible. But in the single cell setting, we usually think of sample size as the number of cells rather than the number of individuals. So in terms of cells in single cell data, usually per individual, you will have thousands of cells. So in that regard, the sample size issue is slightly less of a concern for single cell.
But a bit follow up on that. So if we consider a cell as an independent sample, that won't be a bad interpretation, because generally the cells are from the same sample of origin. So that's why we have suitable methods in single cell analysis, right?
Right. I completely agree with this point. So there is a confusion in the field right now. Actually, you pointed out. So CS4 and CSNet are actually trying to estimate 2 different types of network. With CSNet, we are estimating the network from sample to sample, like from one person to the other person. But in CS4, I'm using cell as a unit. So the network is from cell to cell. So these are 2 different types of network that I completely agree. But with single cell data nowadays, it could be challenging to consider sample heterogeneity there, because usual single cell study only have, say, 10 or 20 subjects. You may have thousands of cells per subject, but subject level, there's only 10 or 20. So with 10 or 20, it goes back to your original question. This very limited sample size make it hard to infer anything basically at a sample level.
Thank you.
That's fair.
Hi, Dr. Su. Thank you for your talk. It's really, really clear walkthrough and really like the method part. I have 2 questions. First is for the CS core research. You talk about the CS core identify upgraded among the COVID-19. Because if I look the picture, I feel like all of the genes are upregulated. Like how, like do you decide it like it's the interferon stimulated genes are up regulated, but not other genes? Because you can see here the heat map is all the genes. Not all, but most of the genes are upregulated.
Mm-hmm. Yeah, thanks for the question. So, maybe let me take a step back. When we do this analysis, we didn't actually start with these genes, but rather we look at all the gene pairs and the entire co-expression network, and we ask, what are the gene pairs that have the most significant changes in co-expression? So that's how we find them. So when I zoom in on all these gene pair that we find to be significantly dysregulated, it would seems like every gene here is very significant, and how do you focus on these these few gene sets? But actually we select them from a much larger set, and this selected set is selected based on the criteria of significant differential co-expression.
Thank you.
Yeah,
And and then my second question is like for CSCO, do you try to use it in like ataxic data? So like we can group the peaks and see if like, for example, some of the peaks are related to one motif or some functions. Can I also try these packages to or ataxic data? That's my question. Thank you.
Yeah, that's a very great question. I'm actually working on this right now. With single cell multi-omic data where you have measurement of, but I think ataxic is the same. So you have like, instead of using the expression measurement model on gene, you can use it on peak. There is some recent paper on Nature Methods saying that single cell ataxic data can be modeled similarly as single cell RNA-seq data. Like you should use a Poisson model. It's a similar type of statistical modeling strategy. So given that as a foundation, I do not see any difference in modeling the network either within genes, between genes and peak, or we can also do peak and peak.
I see. Thank you.
Thanks. Yeah.
Do you think it would be possible to rank or score the genes that you collect using? But if you collect gene sets, is it possible to maybe rank or score them?
Yeah. So the question was, is it possible to rank or score the genes based on the co-expression network? Actually, I guess. Yeah, that's something people do. So when you get a large network, sometimes it's hard to make sense of it. So people compute some summary statistic or like summary of the network. And I think degree is the idea that you just described. Like you can count how many genes is this gene connected to, and we can use that type of summary to rank the genes in terms of how important they are. I imagine a gene with large degree is thought to be like a hub node of the network, and it could be more interesting and important to study. Yeah, I think that's something definitely doable.
Hi, Dr. Xu. This is Liang. I really enjoyed your presentation. I do have two questions. So the first one is my understanding is that you used some, you estimated some kind of correlation instead of partial correlation to map to the network, right? So if so, do you have any insights about the partial correlation? The second one is for the CSNet. Is there any restriction on the N to P ratio? Like how does it work for more N to P ratios?
Yes, those are very good questions. So the first question was on partial correlation. Yeah, definitely. Whenever you estimate correlation, people ask about partial correlation. I think the main challenge here is that for single cell data, maybe this page, that is so sparse and hard to model. The usual partial correlation requires some parametric assumption, or you can also run all these graphical lasso type of approach. But then again, it could suffer from similar confounding issue that we just described, because you are not properly normalized for sequencing that. But definitely there are work in single cell field that on that. I think some from Jessica Lee's group on estimating partial correlation. We didn't do a head-to-head comparison here because it's hard to compare correlation with partial correlation.
Right.
But I think that's still an open field to be worked on. And the second question was on the asymptotics, like the convergence rate was sufficient. Let's go to the slide. Do I have it on the slide? Yeah, so I think we basically put a similar condition as the classical covariance estimation literature from Bickel and Lavina starting 2008, I think. So in their setting, I think it was a while ago when I did proof, but it's basically we're asking P to not grow too fast compared to the sample size n, but I do not have that exact number, like the order in my mind right now, but this is all in the paper.
Okay, thank you.
Okay, well, I think we have a very interesting seminar event, and thank you, Dr. Su for the wonderful talk. Hopefully you have a great rest of the visit.
Yeah, thank you all for the great questions.
Okay. Bye, everyone.